definitely not be on The Voice. Not a good singer. Okay, are we ready? Let me get my pollev.com up. I mean, my poll everywhere up so I can see what everybody's voting. First question is, a segment of DNA that codes for a functional product is A or N. This again should be review. Maybe not, based on your votes. I have votes for A, B, and C. The correct response is B. That is a gene. A gene is a segment of DNA that codes for a specific protein. We can know the sequence of genetic material all we want, but we will have no idea what the function of that gene until it makes the protein, and the protein actually does the job. So this is just a picture that I found that's kind of cool, because one of the things that you need to know actually is that the bacterial chromosome is just one circular piece of DNA, as opposed to linear chromosomes like in eukaryotes. And this one just shows the location of the genes that have been mapped out on this particular bacteria, which is kind of cool. So you can see this one here codes for, it's the gene that codes for the protein catalase, which is an enzyme that allows it to break down certain things. There's a protein for iron uptake. There's a protein that allows it to sense chemotaxis, which is to move towards a gradient for a chemical. There's a gene that has been identified for flagella biosynthesis and assembly. So I just wanted to just kind of hopefully put it in a format that you could understand and go, oh, okay, now I understand what a gene is. So it's the segment of DNA that contains the sequence or the recipe that codes for the protein product that's going to actually carry out the job for that gene, which is of course made at a ribosome. One of the answer keys that I posted for chapter 4 has the organelles and their functions. That should again be review from your previous chapters, but it is, you do need to know that still for this class. Let me clear my responses. We'll move on to 5. Oh wait, somebody Skyped something. I need to see if there's a question. Oh, no, okay. It's just an answer. Okay. Sorry, my nose is tickling again. I hate this. Which process converts one parental double-stranded DNA molecule into 2 identical offspring molecules? There's the different processes for protein synthesis, and then there's the process for copying the genetic material. I don't want to say the word because I'll give it away. Most of you said replication. That is correct. So DNA replication is the copying of all of the genetic material to make a new cell. Transcription is for copying the DNA into RNA to make a protein, and then translation is copying that RNA message into the protein product. I think we have a slide where that comes up later again, so we'll talk about it more and have that picture for it. But this is the process of DNA replication. And in the chapter 6 through 8 need to know PDF, which is on Canvas and modules that I added in the week 2 material, it has a table that has the main enzymes, and there's actually a few extras in that table that you don't need to know. So I really focus on the ones that you really need to know in your group discussions Word document that I posted today, and then also in the study guide review video that I posted. All right. Next question, number 8. In DNA replication, the newly added nucleotide is joined to the growing DNA strand by which enzyme? All right, most of you say A, which is good. That is DNA polymerase. So DNA polymerase adds the new nucleotides in DNA replication RNA polymerase is going to add the RNA nucleotides that are going to form the messenger RNA message from the DNA that's carrying that sequence out to the ribosome to make the protein. So this diagram is straight from your chapter 8 PowerPoint in Canvas, and I basically just highlighted the DNA. These aren't different DNA polymerase, but it's just showing different steps of DNA polymerase. So I just highlighted where they are in each location so that you could understand the different functions. So you have helicase, that is the enzyme that unwinds the DNA. You have DNA polymerase that adds the new bases, forming the new strand. There's a primase, which puts down RNA primers that, especially on the lagging strand that forms the Okazaki fragments, the primase has to get it started with the RNA primer. Then DNA polymerase can come back in. Then you have these little breaks in the sequence, which are called Okazaki fragments on the lagging strand. And DNA ligase is the enzyme that is going to seal those. So those are the main ones that you need to know for DNA replication. All right, 11. Which one of the following enzymes joins the DNA strands, the Okazaki fragments, and mends the gaps in the DNA backbone during excision repair? If you were listening, you got that one easy. So most of you said DNA ligase. That is correct. So DNA ligase not only seals the breaks between the Okazaki fragments during DNA replication, but it is also responsible for DNA repair. We have a whole bunch of proteins that are, I like to call them quality control for the DNA, and they read along it and check for breaks and make sure that the bases are complementary paired correctly. And DNA ligase also seals those breaks. This is just that same picture. I just highlighted the DNA ligase. So DNA ligase seals or glues back together the fragments that are called the Okazaki fragments, and then they're also called the discontinuous fragments on the lagging strand. All right, number 14. What carries the coded information for making specific proteins from DNA to ribosomes? The carrier. All right, good. Most of you said mRNA. That is correct. mRNA stands for messenger RNA, and it is the copy, basically, of the DNA sequence. So that's what is copied in the nucleus. In eukaryotes and prokaryotes, they don't have a nucleus, but it's still, the DNA is still copied into messenger RNA before it goes to a ribosome to be translated into the protein. So the messenger RNA, I'm just actually going to type this in here. Let me make sure I don't have it. Okay, yeah. I'm going to type this in here real quick, and then I'll go on to the next slide. The messenger RNA is the copy of the sequence in one gene. So when we were talking about genes earlier, that's what we're talking about. It's a portion. So the DNA strand is all one continuous piece, but there's just one little section that corresponds to a gene. That part is copied into a messenger RNA that codes for the whole entire protein. What you should also know is that ribosomal RNA and transfer RNA are also transcribed from the DNA sequence. These are considered, they call them non-structural genes. They're genes that don't actually code for proteins, but they are pieces of RNA that have a function. So the DNA codes for, so this would be a copy of the DNA gene that codes for ribosome, for part of the ribosome. Because part of the ribosome is actually pieces of RNA, which is why it's called ribosomal RNA. And then the tRNA is also coded for by a gene, not for ribosomes, but for the RNA that carries the amino acids to the ribosome. hopefully that's kind of review. If you're confused about that, that might be one of the things that you can ask me about, or go back and review the videos and whatnot. But let me go on to this next one. Somebody said something, so I'm going to check and see. This is what I was referring to earlier. So the term gene expression just means protein synthesis. So if a gene is, like I said, if a gene is not making its protein product, then we don't even know what its function is. So we call gene expression protein synthesis because it's actually making the protein product that carries out the function of that gene. Some genes are what we refer to as constitutive, which means that we need those proteins all the time in the same amount. And then some of them are not, and they are dependent on environmental conditions. We don't want to make proteins that we don't need. And so sometimes genes are what we call turned on when the protein product's actually being made. And then we say a gene's turned off if the protein product is not being made. But this is the central dogma that I was referring to. So the DNA contains the genetic sequence. The part that codes for a specific messenger RNA would be the gene. And then that messenger RNA carries that copy of the DNA sequence out to the ribosome, which I have listed here. And the ribosome will read the sequence of the messenger RNA, and each 3 bases correspond to a specific amino acid. So those amino acids are brought in, and that builds the protein product. Hopefully that's working. I am, if anybody wants to type this in there to let anybody know, I am recording this session, so if anybody's having trouble getting in, they can watch the recording of it. I didn't record it in Loom. Oh, I forgot to start. I was going to start Loom. That's okay. I'll worry about that later. Okay, which molecules recognize specific codons and transport amino acids to translate those specific codons? So I was just talking about this. I did download a Skype recording, and I tried to upload it to YouTube, and it was processing forever, so I'm not sure if it did it yet, but that was for the Clicker 3 PowerPoint. So if I get that video link created, I will share that. It's in the Skype history, but. All right, everybody says tRNA. That is correct. Transfer RNA is the piece of RNA that kind of looks like this little blue thing here. And what it has is its complementary sequence on one end that corresponds to the codon in the messenger RNA. So the messenger RNA contains the codon that is from the genetic code chart that you probably learned, that has which 3 bases corresponds to which amino acids. And then the transfer RNA has the complementary bases, and on the other end, it's carrying the specific amino acid that that piece of transfer RNA always carries. Okay, the group of 3 nucleotides on messenger RNA is called A or AN. Oh, now I'm off of my, I was answering her question, so I got off of the pull EV. Most of you said A, codon. That is good. So the codon is on the messenger RNA. The anticodon is on the transfer RNA. So here's the codon. The messenger RNA rides along the ribosome, the small subunit of the ribosome, like a train riding on a train track. And it binds to the ribosome because of the ribosomal RNA that's part of the ribosome that has the complementary bases. And then the anticodon on the transfer RNA will hydrogen bond with the codon on the messenger RNA, and that's how it knows which amino acid comes next in the amino acid sequence for the protein. Okay, next one, 23. Ionizing radiation causes hate that students are having trouble getting in, but can't do anything about it. So, okay. Most of you said insertions and deletions. Oh God, we're all over the place on this one. It is DNA breaks. So ionizing radiation is the end of the electromagnetic spectrum that are the highest energy waves. And I actually have a video linked here. When you download the PDF, you should be able to click on that video that talks about the different types of radiation. Whoops, I don't have another slide for this one. But the ionizing radiation is about down to just before UV light. So X-rays and gamma rays would be ionizing radiation. They have so much energy that they actually break the DNA strand. The non-ionizing radiation can cause damage to the DNA, but it doesn't actually break it. So it doesn't have enough energy to break it, but it can cause mutations also. Things like X-rays and UV light. Okay, what is the most common type of mutation that involves single base pairs? I don't really go into the specific types of mutations on your test. That I don't focus on the different types of mutations. However, because of the fact that antibiotic resistance often occurs due to mutations in the bacterial genome, whether in the chromosomal DNA or whether in the plasmid DNA. It's just, that's why it kind of comes up again in this chapter. But a base substitution is the most common type of mutation that involves single base pairs. And that is because sometimes there are chemical modifications to the bases on the DNA that cause them to base pair incorrectly. And then when it's copied again, it now has the wrong base at that particular location. So I have a little section in here on that. This is a video that actually discusses the process of development of antibiotic resistance in bacteria. Somebody's not muted. I hear a baby crying. There we go. Now we're muted, I think. Okay. So if you download that again, you can click on that to learn more about that also. This largely occurs in the plasmids, which are the smaller circular pieces of DNA separate from the larger circular chromosomal DNA. And the reason for that is just because they're so small that they mutate much more quickly when they're replicating. However, it can also happen in the chromosomal DNA. Now it's freezing and not letting me clear responses. Okay, mutations in which one or a few nucleotide pairs are deleted or inserted in the DNA are called. I didn't read that very clearly, so hopefully y'all read that. I was trying to clear the votes. Oh, somebody said. It's okay. No worries. Frame shift mutation. So a frame shift mutation occurs when there's insertions or deletions of any number of bases other than a multiple of 3. And a frame shift mutation, what it does is not only change one amino acid in the sequence, but it changes all of the amino acids after that sequence because it shifts that reading frame of the 3 bases. And this is just a diagram showing you that the protein product does not fold correctly, and that is why these mutations can cause different problems or benefits for bacteria. And I think it talks about that in that video that I linked as well for the antibiotic resistance, is they have to go through so many mutations to actually get to one that allows them a benefit, but it's just, it must happen that much because it does occur really quickly. Oh, yeah, I think the poll EV is frozen. It's not letting me clear responses either. Just keep trying. If not, that's fine. Okay, this one section, I think there's 1 or 2 questions on the test about this. So this is something that you need to be familiar with. The process of bacteria passing their genes to other microbes of the same generation is known as, that's kind of confusing the way it's worded, but in other words, not through having offspring, not through creating a new generation. So it's saying like through its brothers and sisters, transferring genetic information, which is horizontal gene transfer. I have videos to this in the need to know document, on the 6 to 8 need to know document, and then I have these same videos linked here. So if you watch them in one, it's the exact same video, but again, like I always say, I'm trying to get everything into every place that a student would look for it. So that is referred to as horizontal DNA transfer, as opposed to vertical would be when the cell makes an offspring. Okay, next one. Transformation is the transfer of DNA from a donor recipient, or a donor to a recipient cell, via which mechanism? This is another section that I have in your 6 through 8 group collaboration tables today, is for you to work through these different types of transformation, transduction, and conjugation. All right, so most of you are saying B, which is correct. That is naked DNA in a solution. If you remember the different, trying to remember, studies or findings that contributed to understanding that DNA was the genetic material, the idea of transformation was one of those, because the smooth strain of bacteria was able to transfer the rough strain of bacteria. I can't remember what they're called. But what happens is a bacteria, once it dies, its membrane will break down and its genetic material will be released into the solution, and new bacteria can take up that genetic material and incorporate it into a plasmid or even into their chromosomal DNA. So this video walks through the process of transformation. These are the 3 that you need to know. One of my students came up with a really good way to remember the transduction one, if you have trouble remembering which is which. And that is that it is a bacteriophage that transfers the genetic material in transduction. A bacteriophage is a virus that infects bacteria. And they kind of look like a little spaceship. So one of my students came up with the idea that transduction sounds like abduction, like you're being abducted by an alien. So if that will help you remember that it's a bacteriophage. So it's actually viral DNA that is being injected in transduction. One of the kind of caveats to that, which is kind of more detailed than you need for this class, is that these viruses can actually take up that genetic material from a different bacteria. So viruses can actually transfer DNA from one species of a bacteria to another species of a bacteria through transduction. Conjugation is when the bacteria can share it with another bacteria that's within the same generation, like I was talking about. And this is what we mean by horizontal gene transfer. So two adjacent bacteria can connect via what we call a sex pilus, the pili, which are these small little finger-like projections that allow them to connect together, and then it can transfer its genetic material. And then transformation is the one that we talked about, where the DNA is actually in solution. So you can see this dead bacteria is releasing its genetic material, and this live bacteria is going to take that up and incorporate the genetic material. Okay, so I think these next ones are kind of what we just answered, but the process in which bacterial DNA is transferred from a donor cell to a recipient cell inside a bacteriophage is called which? If y'all clear your own responses. I know some of you it was working for and some of it wasn't. I don't know why it freezes up. Okay, I don't think it's working now because none of the votes are changing, but that is transduction. That was the abduction one. This again is an animation that goes through that process that I just showed in the picture before. All right, and then finally the last one, the process for gene transfer that requires cell-to-cell contact by a particular type of plasmid is called which? So that again is the conjugation, and this animation walks you through that. The plasmid that it's talking about is a separate piece of small circular piece of DNA that codes for that pilus, which is the little finger-like projection that attaches, and it's referred to as the F plasmid because they call it a fertility pilus, even though it's not fertile or for sex. I don't know why they call it that. But anyways, it is just a projection that attaches one bacteria to the other, and they can transfer the genetic material through there. Okay, so hopefully you found that helpful. Let me